Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman, and here's what I bring to you all from the world of medicine. Breaking the curse of dimensionality in genomic data. More recently, rapid technological advances permit RNA sequencing at the single cell level from thousands of cells in parallel, accelerating progress in the biomedical sciences. But quantifying RNAs from such a tiny material poses great technical challenges. Even small errors in the calculations for a large number of genes can quickly add up so that any useful information is lost among signal noise. Now, a team from the Kyoto University Institute for Advanced Study of Human Biology, or WPI-ASHB, has developed a new mathematical method that can eliminate the noise and thus enable the extraction of clear signals from single-cell RNA sequencing data. The new method successfully decreases random sampling noise in the data to enable a precise and complete understanding of a cell's activity. The research has recently been published in the journal Life Science Alliance. The lead author of the paper, Yusuke Imoto from ASHB, explains, each gene represents a different dimension in RNA sequencing data, which means that tens of thousands of dimensions must be collected across multiple cells and analyzed. Even the slightest noise in one dimension can majorly impact the downstream data analysis, so that potentially important signals are lost. This is why we call this the curse of dimensionality. To break the curse of dimensionality, the Kyoto team has developed a new noise reduction method, or RECODE, standing for Resolution of the Curse of Dimensionality, to remove the random sampling noise from single-cell RNA sequencing data. RECODE applies high-dimensional statistical theories to recover accurate results even for genes expressed at very low levels. First, the team tested their method on data from a broadly well-studied cell population, human peripheral blood. They confirmed that RECODE successfully removes the curse of dimensionality to reveal expression patterns for individual genes close to their expected values. Next, when compared against other state-of-the-art analysis methods, RECODE outperformed the competition by giving much truer representations of gene activation. Moreover, RECODE is simpler to use than other methods, without relying on parameters or using machine learning for the calculations to work. Finally, the team tested RECODE on a complex data set from mouse embryo cells containing many different types of cells with unique gene expression patterns. Whereas other methods blurred the results, RECODE clearly resolved gene expression levels even for rare cell types. How oral drugs absorption by stomach is affected by body posture. Researchers from Johns Hopkins University and Johns Hopkins School of Medicine employed a biomimetic in silico simulator based on the realistic anatomy and morphology of the stomach, a stomach sim, to investigate and quantify the effect of the body posture and stomach motility on drug bioavailability. Stomach contents, motility, and gastric fluid dynamics all play a role in a drug's bioavailability, and stomach contractions can induce pressure and generate complex pill trajectories. This results in varying rates of pill dissolution and non-uniform emptying of the drug into the duodenum, and sometimes gastric dumping in the case of modified release dosage. Together, these issues pose several challenges for the design of drug delivery. In this work, we demonstrate a novel simulation platform that offers the potential for overcoming these limitations, said co-author Rajat Mittal. Our models can generate biorelevant data on drug dissolution that can provide useful and unique insights into the complex physiological processes behind the oral administration of pills. Lowered risk of heart attack or stroke and death using dietary salt substitutes. Dietary salt substitutes lower the risk of heart attack, stroke, and death from all causes and cardiovascular disease, finds a pooled data analysis of the available evidence published online in the journal Heart. The beneficial effects of these substitutes are likely to apply to people all around the world, say the researchers. Salt substitutes in which a proportion of sodium chloride is replaced with potassium chloride are known to help lower blood pressure. The researchers trawled research databases looking for randomized clinical trials published up to the end of August 2021 and reporting on the effects of a salt substitute on blood pressure, cardiovascular health, and early death. They pooled the results of 21 relevant international clinical trials involving nearly 30,000 people carried out in Europe, the Western Pacific region, the Americas, and Southeast Asia. The study periods lasted from 1 month to 5 years. The proportion of sodium chloride in the salt substitutes varied from 33% to 75%, The proportion of potassium ranged from 25% to 65%. The pool data analysis showed that salt substitutes lowered blood pressure in all the participants. The overall reduction in systolic blood pressure was 4.61 mmHg and the overall reduction in diastolic blood pressure was 1.61 mmHg. Reductions in blood pressure seem to be consistent irrespective of geography, age, sex, history of high blood pressure, weight or BMI, baseline blood pressure, and baseline levels of urinary sodium and potassium. A pool data analysis of the results of 5 of those trials involving more than 24,000 participants showed that salt substitutes lowered the risks of early death from any cause by 11%, from cardiovascular disease by 13%, and the risk of heart attack or stroke by 11%. Accurate prediction of diabetes and pre-diabetes by AI plus ECG heart trace. Structural and functional changes in the cardiovascular system occur early on, even before indicative blood glucose changes, and these show up on an ECG heart trace. The researchers therefore wanted to see if machine learning or AI techniques could be used to harness the screening potential of ECG to predict pre-diabetes and type 2 diabetes in people at high risk of the disease. They drew on participants in the diabetes in Sindhi families in Nagpur or DISFIN study, which looked at the genetic basis of type 2 diabetes and other metabolic traits in Sindhi families at high risk of the disease in Nagpur, India. Families with at least one known cause of type 2 diabetes and living in Nagpur, which has a high density of Sindhi people, were enrolled in the study. Participants provided details of their personal and family medical histories, their normal diet, and underwent a full range of blood tests and clinical assessments. The average age was 48 and 61% of them were women. Pre-diabetes and diabetes were identified from the diagnostic criteria specified by the American Diabetes Association. The prevalence of both type 2 diabetes and pre-diabetes was high, around 30% and 14% respectively, and the prevalence of insulin resistance was also high, that is 35%, as was the prevalence of other influential coexisting conditions, that is high blood pressure 51%, obesity around 40%, and disordered blood fats, that is 36%. A standard 12-lead ECG heart trace lasting 10 seconds was done for each of the 1,262 participants included, and 100 unique structural and functional features for each lead were combined for each of the 10,461 single heartbeats recorded to generate a predictive algorithm, or diabetes. Based on the shape and size of the individual heartbeats, the diabetes algorithm quickly detected diabetes and pre-diabetes with an overall accuracy of 97% and a precision of 97%, irrespective of influential factors such as age, gender, and coexisting metabolic disorders. Important ECG features consistently match the known biological triggers underpinning cardiac changes that are typical of diabetes and pre-diabetes. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.